Let's take a look at DNA mutations. Mutations are just a change in the DNA nucleotide sequence, and they can be caused by a couple different things. Most commonly, they are caused by mistakes during DNA replication, and these are just random mistakes that happen, and they're not caused by any outside force. And then the other cause of DNA mutations would be damage to the DNA that can be caused by exposure to things like radiation. Radiation would be things like exposure to UV radiation from the sun, or maybe radiation from X-rays, like too many X-rays being taken, or maybe like gamma radiation from like radioactive things. And then you can also have DNA damage from being exposed to certain nasty chemicals as well. The types of mutations that are most common are called point mutations. And point mutations are where you're just changing a small part of the DNA, usually just one nucleotide pair. And we've got a couple different types of point mutations. So these can be base substitutions, insertions, or deletions. So let's just take a look at these three different types of point mutations. So first we have a base substitution. So in our double-stranded DNA here, we have a C and a G nucleotide pair, but maybe we have a mistake in DNA replication, and instead of a C and a G, now we've got a T and an A, where there should be a C and a G. So that could cause a base substitution mutation. And then over here on the top right, we've got, up here on the very top, we've got a single strand of DNA, and we've just simplified it instead of having the double strand, just the single strand to make it easier to see. And we've had an insertion. So we've inserted an extra T in the DNA. And then on the bottom, we've had a deletion. So here we've deleted this A nucleotide, and on the bottom here you can see that that A nucleotide is now gone. All right, so these base substitutions where we swapped out maybe the CG for a TA in the DNA, that can cause a couple different types of mutations. So that could cause a silent mutation, a missense mutation, or a nonsense mutation. So make sure you know the difference between these three. So a silent mutation, what happens there is that you do have a change in the DNA sequence, but you end up coding for the same amino acid. And why that happens is that we'll see a little bit later, but there's some redundancy in the genetic code. So we do have some sequences that they're a little bit different in the DNA sequence, but they do end up coding for the same exact amino acid. So a silent mutation, you end up coding for the same amino acid even though you have changed the nucleotide sequence of the DNA. So that's why they call it a silent mutation, because it ends up not changing the protein at all that's coded for. A missense mutation, you end up coding for a different amino acid than what's supposed to be there. So that may or may not change the function of the protein. It depends on what amino acid gets put into the protein. That's being coded for, that's different. And also where in the protein that amino acid is located. So who knows what will happen with that one. And then a nonsense mutation, this one is going to code for a stop codon where there's not supposed to be one. So what happens is that as you're making the protein, you end up stop making it too soon. So the protein ends up being too short and you're missing the rest of the protein. So a lot of times this one ends up being the worst one to have because part of your protein is missing. And then with the insertions and deletions, those point mutations end up causing a frame shift mutation. So we end up shifting the reading frame. Our groupings of 3 nucleotides will show you what we mean in just a minute. And that will shift what amino acids we're actually coding for. All right, so let's take a look at these silent, missense, and nonsense mutations. So we've got a single strand of DNA here, just to simplify things. And then here's the amino acids that are being coded for. So we're going to be looking at this second amino acid here. And notice that we've got a grouping of 3 in the DNA. We call that a triplet in the DNA. And that triplet will end up coding for a particular amino acid or a stop codon. And we can see this would be what we're supposed to be coding for. This is the normal on the left. And then you can see that where we have this valine at the second position, we've got a GTA in the DNA. And then you can see over here on the right, we've had a mutation. So we've swapped out the A, and now we've got a T there. So instead of GTA, we're reading GTT. And so with that base substitution, we end up causing a silent mutation because we end up coding for the same exact amino acid. So even though our nucleotide sequence has changed in the DNA, so we have GTA and now we have GTT, we end up coding for the same exact amino acids, so a valine to a valine, and so that would be a silent mutation. So we actually haven't changed the protein at all, so it's going to have the same function. So that would probably be the best of the mutations to have because you're not going to change the protein function. All right, our next one, we've got a missense mutation. And in this one, we're going to focus on our third amino acid. We've got a proline here. And in our DNA, we've got CCC for our nucleotide triplet. And if we look over here on the right, we've had a mutation where that first C has changed to an A. So now we have ACC. And ACC changes our amino acid that we're going to be coding for, and that changes to threonine. So this would be a missense mutation because instead of coding for proline, we're going to be coding for a different amino acid, threonine. So that's a missense mutation. And then our last one, we've got a nonsense mutation here. So we're going to be looking at the last amino acid here. So in the DNA, we've got TAC as our triplet sequence here, coding for tyrosine. But we've had a mutation in our last letter here. So instead of TAC, now we've got TAG. And then that will actually end up coding for a stop codon. And we end up not putting an amino acid there. And that just says, hey, that's the end of your protein. Stop making the protein. And so we end up with a protein that is too short. So this would be a nonsense mutation where we would end up coding for a stop codon where there shouldn't be one. Okay, and then the last type of point mutation we have are these insertions and deletions. So these will cause a frame shift mutation if we end up inserting or deleting 1 or 2 nucleotides in our DNA. So this one, we're going to be deleting the TA in our DNA. And this one over here on the left would be the normal, what we should have. So this should be our sequence of amino acids we should be coding for. But we've deleted the T and A. So when we do our groupings of 3, when we translate the messenger RNA, when we read that code on the messenger RNA to figure out what amino acid to put in what order, we're going to be shifting our reading frame. So our AGC is okay, so we're still going to code for serine. But our next grouping of 3, we should have a GUA. But because we've deleted that TA up here in the DNA, we're missing that UA in the messenger RNA, so we're just going to grab the next 2 letters, And so we'll group GCC together. And you can see that that's going to code for the amino acid alanine instead of valine. And so our next grouping of 3 is also going to be off. We're also going to be shifting the reading frame there. So instead of CCC, we've got CUA, and that'll code for leucine. And then our next amino acid is also going to be off. So instead of UAC coding for tyrosine, we've got CUU coding for another leucine. So this would be a frame shift mutation where we shift our reading frame or groupings of 3, and this would be due to a deletion or an insertion of nucleotides in the DNA. Okay, and then just the last thing to mention, mutations can be either bad, good, or neutral. So it just depends. So a lot of times we think mutations are bad, and for good reasons. So some examples of bad mutations. They can cause cancer. They can also cause some other genetic diseases like cystic fibrosis, sickle cell anemia, muscular dystrophy. There's a lot of genetic diseases that are out there that are caused by bad mutations. But some mutations are actually good. So for example, being able to digest lactose in milk and dairy products. There was a mutation that allowed us to be able to do that, and so that was actually a good mutation. And if you're lactose intolerant, you do not have that mutation, and so if you eat dairy products, that probably is not going to be good for your GI tract. There are some people that are resistant to getting HIV, so they've had a mutation that makes them so that it's going to be very hard for them to get HIV and be infected with that. So those would be good mutations. Some mutations are neutral. They're not going to cause any problems. So for example, the silent mutations, they're neutral. Okay, they end up coding for the same amino acid. They're not going to change the protein. Another example of a neutral mutation would be, what if the mutation occurs in a part of the DNA that's not coding for any part of the protein? That's not going to affect the protein at all. Another neutral mutation would be one that doesn't affect the survival of the organism. So for example, in humans, there's different mutations that cause the different eye colors in humans. So having brown eyes versus blue eyes, that doesn't affect your survival. So that would be a neutral mutation.